right, so in this video, we're going to skip all the way down here and we're going to list the various characteristics of an antigen. We're going to describe the types of T-cell antigen receptors. We're going to outline the steps of T-cell development. We're going to describe the major T-cell types and their functions. Okay, so this conversation that we're having today is all about adaptive acquired immunity. Okay. So the adaptive defense system is a more specific response to pathogens that may enter our body. We consider it the third line of defense as it attacks specific foreign antigens. It takes a little bit of time to react. We have to develop this system, and the cells that play a huge role in the system has to be introduced to the pathogen initially and then develop kind of cells that can be better prepared to attack it if it encountered it later. Okay, so over time it builds a specific response to eliminate the specific antigen, creating what is known as immunological memory. Okay, adaptive responses are carried out by lymphocytes and macrophages that can recognize specific antigens. There's two types of adaptive defenses. We have our cellular immune response, which are performed by the immune cells. And then we have our humoral immune response, which is performed by antibodies. And so as a recap, the pathogen will encounter the first line of defense, and if the integrity of that defense is breached, and the pathogen makes its way in, it will either encounter the second line of defense, which we talked about in the earlier video, or it can be addressed by a third line of defense, which includes the mechanisms of adaptive immunity that we're about to talk about now. All right, so let's talk about the cells that we find in adaptive immunity. We have both T cells and B cells. These are lymphocytes, and they, like all of our blood cells, whether it's red blood cells or white blood cells, they originate in the red bone marrow. So T cells and B cells both originate in the red bone marrow. Where they differentiate or mature rather are in two different places. So the T cells mature in the thymus. The B cells mature in the red bone marrow. So if it helps you remember, T cells with thymus, T thymus, okay? B cells with red bone marrow, okay? When we look at how they're dispersed throughout the body, our T cells are going to be found in our lymphatic tissues, and then they represent roughly 70 to 80% of our lymphocytes that are circulating in our blood. Whereas our B cells, they're also found in our lymphatic tissues, but there's not as much circulate in the blood, roughly 20 to 30%, okay? The primary functions of our T cells include providing a cellular response in which the T cell interacts directly with the antigens or antigen-bearing agents to destroy them, whereas the B cells provide a humoral response where they interact indirectly. Basically, they produce antibodies that destroy the antigens or antigen-bearing agents. So let's look at cell-mediated immunity, our T cells. A lymphocyte has to be activated before it can respond to the presence of an antigen. T cell activation requires that the antigenic fragments be attached to an antigen-presenting cell, or APC. And so basically an APC will phagocytize the antigen and digest it and display its fragments on the cell's surface, okay? Typically it's going to be bound to a protein that is a part of the major histocompatibility complex. So typically you're going to see this antigen presenting cell with another protein on the surface called an MHC, okay? When specific T cells encounters an APC displaying foreign antigen fragments bound to the MHC proteins, it can now recognize the antigen as foreign. And so at this point, our T cell is now activated. T cells can secrete polypeptides called cytokines, which can enhance some cellular responses to antigens. Now we have different types of T cells. We have helper T cells, which are going to activate other cells by secreting cytokines, and it's going to stimulate B cells to produce antibodies, okay? It also can stimulate some of the activities of our cytotoxic T cells. We have cytotoxic T cells, which are going to attack the virally infected or cancerous cells. We have memory T cells, which are going to provide future immune protection. And then we have regulatory T cells, which are going to suppress the immune responses after the pathogen has been eliminated, which lowers the chance of developing an autoimmune disorder. So this illustration just describes, or illustrates rather, what I was explaining on the previous slide. So let's go through those steps again. So first we're going to have an antigen presenting cell. So we're going to have a cell that has been identified as an abnormal, non-self pathogenic cell. And the macrophage is going to engulf it. It's going to phagocytize it and take it into its plasma membrane. There are going to be lysosomes and other enzymes present that are going to cause the breakdown of the pathogenic cell into fragments. Excuse me. And those fragments are going to be displayed all around the surface, or the membrane of that macrophage cell. So you can see the fragments here, the fragments here, and the fragments here, okay? A helper T cell is going to come in and it's going to bind to that antigen that's located on the surface of that macrophage, okay? And when it does that, it's going to cause the helper T cells to proliferate. And so when they proliferate, the proliferating T cells will start to secrete cytokines. One cytokine that it's going to release is called interleukin-2. Okay, so we have interleukin-2. The presence of interleukin-2 is going to cause our cytotoxic T cells to proliferate and differentiate. Okay, so basically the interleukin-2 is going to lead to the activation of those T cells. Antigen presentation is going to be vital for the activation of T cells. T cells respond only to processed fragments of antigens displayed on surfaces of cells by the major histocompatibility complex proteins. And there are two different classes of MHC proteins. We have MHC class 1 proteins, which are found on all body cells, cells that feature a nucleus, okay? And they have the ability to bind and display self or non-self antigens. Cytotoxic T cells recognize and bind to class 1 MHC antigen complex. Now, self antigens do not trigger an immune response, but foreign antigens will activate the cytotoxic T cells. We also have MHC class 2 proteins that are only found on the surface of macrophages and B cells. They act as an antigen presenting cell for helper T cells, and once activated, the cells will start their immune response, typically by secreting cytokines that can lead to the proliferation and activation of our T and B cells. So with this diagram, we are going to focus on steps involved with cell-mediated immunity, looking specifically at our T cells, okay? We're going to have pre-existing antibodies that have the ability to bind to the antigen and mark it and opsonize it, okay? The macrophage is going to identify it and engulf it. It's also going to fragment it and bind to the MHC2 complexes that are present on the macrophage surface. Now, the T cell can bind to the MHC2 complex and activate the helper T cells, causing it to proliferate and secrete cytokines, which can either activate and recruit cytotoxic T cells, or it can lead to an inflammatory response. Two things can happen at once, okay? And so once these cytokines are released, again, it can activate our cytotoxic T cells. It can also activate our B lymphocytes as well, Okay, so there's two fates here. Okay, cytotoxic T cells or activate NRB lymphocytes, as you can see here.